Achieving sub-nanometer spatial resolution in material science has historically required the sheer scale and immense power of traditional electron microscopes. Extracting this level of detail carries a heavy physical cost. To be imaged, samples cannot remain in their native state. They must be frozen, desiccated, or locked inside a hard vacuum seal. This rigorous preparation creates an illusion of stasis. By locking molecules in place, we inadvertently force chemists to conceptualize flexible structures, like block copolymers and liposomes, as permanent, rigid architectures. Biological and synthetic systems are actually highly volatile. When suspended in their native aqueous environments, these structures are constantly shifting and exchanging parts. While crystallography and electron microscopy provide perfect structural scaffolding, they remain blind to the kinetic thermodynamics of molecular assembly. The barrier to viewing these materials in their natural state using traditional optical microscopy is the Abbe diffraction limit, which caps maximum resolution at roughly 200 nanometers. Resolving any nanomaterial smaller than this threshold requires engineering techniques that actively circumvent the fundamental wavelength properties of light. Super-resolution microscopy is the disruptive optical solution to this fundamental boundary, achieving 5 to 50 nanometer resolution. Unlike electron microscopy, these techniques operate in operando. Researchers observe materials directly in their native solvents, avoiding the need for freezing or vacuum states. By tagging specific components with fluorophores, researchers can trace individual molecules in real time. This capability allows us to rewrite the foundational kinetic models for DNA, lipids, and supramolecular polymers. We break the diffraction limit using 3 primary techniques: structured illumination, stimulated emission depletion, and single-molecule localization microscopy. Single-molecule localization, which includes methods like STORM and PAINT, solves the blurry ensemble problem created by overlapping light waves. To isolate individual fluorophores from the blur, these methods deploy photo-switchable probes or reversible binding dyes. This creates a stochastic blinking mechanism. The system ensures only a sparse, random subset of molecules emits light. Because flashes are separated by dark space, algorithms map their point spread functions. Software calculates the exact center of each emission, constructing a precise sub-diffraction image. This process transforms a physical optical blur into a precise, navigable mathematical coordinate map down to 5 nanometers. We see the application of this mapping in DNA nanotechnology, where 3D polyhedra serve as programmable molecular breadboards. The DNA PAINT method relies on the transient, reversible hybridization of labeled oligonucleotides, rather than permanently attaching fluorophores to the structure. Short, fluorescent DNA strands continuously bind and unbind from specific docking sites on the origami structure. These highly predictable binding kinetics provide the exact blinking rate required for localization algorithms. This approach yields resolution under 15 nanometers laterally and 24 nanometers axially. This precision allows researchers to perform absolute quantification of binding sites on a nanostructure, counting exact protein copy numbers. DNA PAINT proves that we can resolve complex 3D architectures while simultaneously quantifying their kinetic interactions in real time. This capacity for real-time observation scales to synthetic materials, such as water-soluble BTA supramolecular polymers. A long-standing dogma from static imaging assumed that monomer exchange in these fibers only occurred at the exposed ends or termini of the chain. To test this, researchers designed a 2-color STORM experiment to track monomer exchange in these assemblies over time. The setup involved introducing red-labeled monomers into an aqueous solution of pre-assembled green-labeled polymer chains. If the static assumption were correct, red monomers would only appear at the tips. Instead, the STORM data revealed homogeneous integration. It definitively proved that exchange takes place across the entire backbone simultaneously. The whole structure is volatile. Supramolecular polymers must now be understood as breathing, dynamic entities capable of instantaneous structural reaction rather than static, unbreakable wires. This kinetic volatility is even more critical at the bio-nano interface. the moment synthetic nanoparticles enter biological fluids. Immediately upon injection, a biomolecular corona forms, a complex coating of proteins that dictates how the body reacts. Quantifying how deep these soft proteins penetrate into porous silica nanoparticles is nearly impossible with traditional imaging. Using D-Storm, researchers can track this intrusion in mesoporous silica nanoparticles modified with varying lengths of PEG chains. This comparative data illustrates how modifying the PEG molecular weight alters the depth of protein intrusion. As the molecular weight of the PEG coating increases, the penetration depth of the protein corona drastically decreases. This quantitative data allows nanomedicine engineers to design better cloaking and shielding for targeted drug delivery systems. Super-resolution mapping acts as the ultimate bridge between synthetic material design and biological reality by quantifying the dynamic biointerface. Techniques like STORM, STED, and PAINT bypass the diffraction limit to expose the thermodynamic and kinetic reality of assemblies in motion. However, these methods are not a panacea. They cannot fully replace the physical, atomic density mapping provided by electron or atomic force microscopy. The new frontier of advanced materials characterization is correlative microscopy. This approach merges two distinct imaging modalities, overlaying the grayscale atomic density of the electron microscope with the vibrant kinetic tracking of super-resolution optics. The result is a system that provides the pristine structural scaffolding required for baseline architecture, as well as the dynamic fidelity needed to track behavior. Constructing functional, responsive materials requires intimately knowing both their building blocks and their volatile behaviors. To build the next generation of nanotechnology and precision medicine, we must stop studying materials as static artifacts and begin designing them as